Hello. I decided rather than making a PowerPoint for this, I would rather just make a video for chapter 21 because I'm slow at PowerPoints, but I can talk fast. All right, so with chapter 21, we're dealing with microevolution, which on the handy dandy notes that I've attached for you tell us that means small scale changes in allele frequencies due to natural selection, mutation, gene flow, or genetic drift. Some of those terms we know, some of those terms we don't know, we'll get into in this chapter. But with micro, obviously we're talking small. Allele frequencies meaning how often does that allele show up in a population. So I've put on here with arrows that all of those factors can cause allele frequencies to change, positively or negatively. And then by definition, that's what we mean by evolution. Now, one important point for you to understand on this, and that's why I put it at number 1 here, and I'm showing this slide. Individual organisms do not evolve. You cannot evolve, I cannot evolve in a biological sense. Populations of organisms do evolve. So when we talk about allele frequency, we have to consider in a whole population, not just in you or some scallop growing in the ocean. All right, so individuals do not evolve, populations do. So by extension, then what we mean by evolution, A1, is genetic change in a line of descent over generations of a population. Genetic change in a line of descent over generations of a population. Okay? Well then, let's go back. What is a population? Just to refresh our memory. A population is a group of individuals of the same species, meaning they'll have shared characteristics, living in a given area which have two identifiable characteristics. A, they'll share traits. So those could be morphological, like physical traits. They could share physiological traits like body processes, carrying out photosynthesis, having the ability to camouflage. And they can have behavioral traits like being nocturnal or migrating, things like that. So members of a population will share traits, obviously, and members of a population may have some variation in those traits. The alleles that create those traits may not all be exactly the same. And that variation can be of two types, qualitative and quantitative. Qualitative variation means, like we've been dealing with genetics, either or. You can either roll your tongue or you cannot. You either produce purple flowers or white flowers. That's qualitative variation. Quantitative variation is more like what we have for most traits, where you have a continuum of possibilities, a range of variation. In other words, it's a polygenic trait. So like we've talked about with skin color and human height. And the human population pictured in here, or representatives of the human populations, are demonstrating the variation within skin color. We'll see that sometimes the effects on individuals have an effect in the population. We'll get into that a little bit later. All right, if we take a population, so we've established what a population is, and we think about that each member of the population has alleles for traits, we could toss all of their alleles into a container and we could say we have a pool of alleles. Wait, here's a container right here. So if we consider our class as our population, I could take everybody's alleles in the class, pitch them in here, and we've got a pool of alleles. That pool then, if I draw an allele out, at random, which allele am I going to get? Well, some alleles are going to be more common in our population than others, so the probability is I'm more likely to draw that allele out. So we'll be learning in just a moment how to work with that mathematically and figure out probabilities. All right, the genes or the alleles are not the whole story. Remember that environment can also affect phenotype. Back to skin color. The outward expression of your skin color may also be a function of the amount of time you spend in the sun or don't spend in the sun. That also may be true for other organisms. The height of plants could be a function of where they grow in relationship to shade by other plants. And so being shaded, they may not reach as great a height as other members of their population that get more hours of sunlight. Within an individual, your individual alleles are affected by 6 factors. All right, so an individual's alleles are affected by 6 factors. Number 1, mutation. What alleles an individual has would be affected by mutation. Let's skip over some slides. Here's our variation within populations. Pretty horses. Right, so mutation. Mutation is the only one of these factors that has the ability to create new alleles. The other 5 will only shuffle the deck of existing alleles, if you will. So mutation is the only one that can create new alleles. All right, secondly, an individual's alleles are affected by crossing over. Third factor would be independent assortment. A 4th would be which random gametes meet at fertilization. So all of those are factors we discussed when we talked about meiosis, if you remember. A 5th one would be chromosomal changes in terms of chromosome number or structure. And then a 6th one would be migration, which is another term for what we'll see soon called gene flow, whether organisms move in and out of a population. All right, so mutation can cause variation. It's random. Mutation is constantly occurring. Gene flow is due to individuals moving in and out of a population, which therefore means they bring their alleles with them or take their alleles away with them. So you're always going to have populations that have some mixing, some genetic mixing when individuals come in and join their population. And that's going to be true in nature always. And it's becoming more and more true in human populations, right? I think we had talked about the possibilities for another U, and we had realized the numbers are quite huge. If you combine all of these factors that affect your individual allele makeup, 10 to the 600th power possible combinations for each gamete. Okay, that's a lot. All right, let's think about whether or not the allele frequencies are changing. So let's assume that if we have something we call genetic equilibrium, and that's at the very, actually we're going to define it at the top of the next page. I want to define something else first, sorry. Allele frequency is exactly what the name tells you. It is how often an allele appears in a population's gene pool. Allele frequency is how often an allele appears in a population's gene pool. Okay, so at the top of the next page, in genetic equilibrium, what do we mean? We mean that the allele frequencies are fixed or unchanging from one generation to the next. So for example, let's just say we have allele big A and allele little a. And let's say that allele big A, which would appear to be the dominant allele, has a 70% frequency in the population, and allele little a has a 30% frequency in the population. If our population is in genetic equilibrium, the next generation, those numbers will be the same. The next generation, those numbers will be the same. Well, what does that mean? That means that population is not evolving, because just a minute ago we said evolution is genetic change in a line of descent from generation to generation. So if these frequencies don't change, this population is not evolving. Now, does that happen in real life? Actually, not very often, but it's going to be helpful to us to consider, if that were the case, what conditions would be going on? What could we assume in a population that is not evolving? So we're going to have 5 factors that we can assume if our population is not evolving. Say 5 conditions that are being met. Number 1, there are no mutations. Well, right off the bat, we know that's not going to happen. Of course, mutations are always occurring, but this is a hypothetical, okay? So one condition, no mutations. The second condition is that you have a very large population. Why is that important? Well, think about flipping a coin. If I gave each of you a coin and told you to flip it 10 times, we expect that you get 5 and 5, heads and tails. But because 10 is a small number, it would be very possible that each of you could get different outcomes. If I told you to flip it 1,000 times, after you stopped with the eye rolling and said, oh my God, I can't believe you're making me do that, what's the likelihood? We're going to be much closer to 500, 500 if I make you do it 1,000 times. So a large population means that you're more likely to have statistical results like you expect. And allele frequencies would be unchanging more often if your population is large. All right, a third condition is that your population is isolated from other populations. We can't have any of this genetic mixing going on. Otherwise, that would cause allele frequencies to change. And we've said no. Our conditions are saying no evolution's going on here. So far, no mutation, large population, population is isolated from other populations. Number 4, the gene that we're considering, okay, gene A, has no effect on survival or reproduction. Well, okay, we could think of examples of genes that have no effect on survival or reproduction. Earlobes and tongue rolling come to mind. But most genes are going to have some effect on survival and therefore reproduction. So again, that's why this is hypothetical. The 5th condition that has to be met is that all mating is random. So 2 members of the population meet, they'll mate. There is no courtship behavior. There's no selection because the females choose which males they want to meet. Nope, if your population's not evolving, that's not going on. All mating is random. So again, these are 5 conditions in a population not evolving, but that's not going to really be the case in most real world examples. So let's choose an example. Let me end my slideshow. Because I need to look at a picture. Here. Here's a field with some wildflowers in it. All right, so we're going to have a population of wildflowers, and instead of alleles big A and little a, we're going to use alleles big R and little r. All right, so with our wildflowers, with alleles big R, which stands for red, and little r, which stands for white. All right, let's say that we have a population of 500 wildflowers is our total. And in our population of 500 of them, 480 of them are red, and 20 of them are white. Now, we have to go back to genetics a little bit. Remember that we're going to use Mendelian genetics with this. We're going to say that the red allele is completely dominant over the white allele. This is simple dominant inheritance. Therefore, the red flowers could be homozygous dominant, or they could be heterozygous. We don't have any way to know. We can't look at these pretty wildflowers in the field and know whether they're heterozygous or homozygous dominant. We just know genetically speaking they can be one or the other. So we have to think about this. Let's say that we are given in our population, we know if we have 500 individuals, then that gives us 1,000 alleles. Ooh, how did I get to that? Okay, it's a single trait. Each individual will inherit 2 alleles for that trait, one from each parent. So therefore, 500 times 2 gives us 1,000 alleles in our gene pool. Sure hope this is filming so that you can see all of this. I'll try to stay in this corner. All right, let's say that I'm going to tell you 320 of our reds are going to be homozygous dominant. 320. So that means they have 2 big R alleles. Change how I set this up. Okay. Because you're quick with math, I bet you're already thinking, well then out of our 480, that must mean that we have 160 that are heterozygous for red. That means they only have 1 big R allele. So that tells me if I add up the 2, I get 2 copies of the big R allele from the homozygous dominants. I get 1 copy of the allele from the heterozygotes. So I have 800 alleles in my gene pool. Big R's. So finding the allele frequency, I'll bet you're already a step ahead of me. 800 for that allele out of our total gene pool of alleles is 1,000. So that means that 0.8 or 80% is the frequency, allele frequency. For big R. Okay. So far so good, I hope. Now, what do you think the allele frequency for little r would be? Well, we can do the math. You could go ahead and think in your head, all right, so if we have 20 individuals that are little r, each of them has 2 copies of the little r allele, so that's 40. We had 160 a minute ago who were heterozygous. they have one copy of the little r allele. Therefore, 200 little r alleles in our gene pool, and so mathematically, our allele frequency for the little r allele is 20% or 0.2. You probably figured out already that the two allele frequencies have to equal up to 100%. So if you know one, you can automatically subtract and figure out the other. Ah, okay. This is going to be easy. All right. Let's look in the middle of your second page of notes, thinking about mutations one more time. Mutation rate. Each gene has a probability of mutation occurring during replication. That's what we mean by mutation rate. The probability of mutation occurring during replication. On average, 10 to the 5th to 10 to the 6th per gene. Those are pretty typical mutation rates taken for genes. Some genes mutate more often than that. Some mutate much more slowly than that. Now, let's think about the effects of mutations. Mutations could be beneficial. They could lead to a trait that increases your survivability. Mutations could be lethal because changing the protein oftentimes is not a good thing, or they could be neutral. It could have no effect. Remember our silent mutations, right? So which of those actually would be the cause of evolution? Well, it should come as no surprise to you that really evolution is the accumulation of the beneficial mutations. So we may be accustomed to thinking all mutation is bad. No, not true. That's how we have so much variety. All right, so a couple of scientists developed a mathematical equation to describe what's going on when a population is not evolving, and that's called the Hardy-Weinberg rule. The Hardy-Weinberg rule uses some of the assumptions we just talked about. So it assumes no mutations, it assumes large population, it assumes isolated population, it assumes random mating, and it assumes that survival and reproduction is equal for all individuals. Remember, we said that the genes aren't going to have any effect on survival and reproduction. So everybody is equally likely to survive and reproduce. Well, we know that's not true in the real world, but in this situation it is. That's what Hardy-Weinberg works on. So if those assumptions are met, then the allele frequencies will remain the same. So with our field of wildflowers, the next generation of wildflowers, the red allele will still be 80% and the white allele will be 20%. And then the next generation, 80%, 20%. So we could say that that population is in genetic equilibrium, or we can also call it Hardy-Weinberg equilibrium. Now, the Hardy-Weinberg equations are on the next page. So there are two basic equations that we need to understand. One of those equations uses the letters P and Q. Okay. So this first equation, P stands for the allele frequency of the dominant allele. So in other words, just a minute ago with our wildflowers, the allele frequency of the dominant allele was the red one, and so that was 0.8. So in this situation, P would be 0.8. Okay. So Q is going to be the allele frequency of the recessive. So in our scenario a few minutes ago, that would have been 0.2. Q would have been 0.2. Okay. So if we understand what P and Q mean, then what this equation does is take those frequencies and figure out the frequencies of the different genotypes. P squared is the homozygous dominant genotype. Q squared is the homozygous recessive. And 2PQ represents the heterozygotes. Let's look at a Punnett square to see why that is. If we had a cross, a monohybrid cross, between two individuals that were heterozygous. So back to simple Mendelian genetics, and I'm not going to fill up the box for a reason. Okay. If we know that the allele frequency for the dominant allele, big R, was 0.8, then we can say that this gamete providing big R and this gamete providing big R, then 0.64 is the combination of those two, giving us a homozygous dominant phenotype. Okay, 0.8 times 0.8. If we know that little r is 0.2, then down here, 0.2 times 0.2 is 0.04. That's giving us the frequency of homozygous recessive. So that's Q squared. And then you can obviously see there are two ways to be heterozygous. So 0.2 times 0.8, 0.16 is P times Q. 0.16 is P times Q here. But there are two heterozygotes produced. So that's why we have a number 2 in front of it. Okay, so what's this telling us? These are population or genotype frequencies. So we'll be able to solve problems that will ask you what percent of the population would we expect to be heterozygous? What percent of the population would we expect to be homozygous dominant? You'll be given some numbers, obviously, in the problem. But if it says population or genotypes are mentioned, like this, then you're going to use this equation. All right, the second equation, much easier, and we actually worked with it a minute ago before you even realized that you knew it. And that equation tells us that the combined allele frequencies of our two alleles has to equal 100%. In other words, it has to equal 1. P and Q are allele frequencies. Okay? So when it asks you the frequency of the big R allele, this is the equation that will help you figure that out. Genotype or population frequencies uses this one. Allele frequencies uses this one. Okay. Let's do an example of two. So in a population with two alleles, big B and little b,